Hello, dear students. Welcome to online classes of Cell Biology and Biotechnology, Paper 5. I'm Miss Kavya Shree, Assistant Professor, Department of Zoology, Histones College, Bantwal. So, we are starting with a new topic from Unit 3, that is mitosis. Okay. So, before we study the stages of mitosis, it is very important to know about cell cycle and cell division. So we know that cellular organization is a characteristic feature of each and every living organism, right? So if you are calling a particular thing as living organism, this is a must that it is found of a cell, right? So the cell has different properties. So main two properties are it will grow and divide. For example, in this diagram, see if you consider this as a cell, okay, it has capacity to grow in its size. That means actually in the cell the cytoplasmic content will be increased, and the grown cell, which is divided into two equal daughter cells. Clear? Okay, so what happens here? Initially it increases in its cytoplasmic content of a particular cell, which is known as cell growth. Okay, whereas when that particular grown cell, which is divided into two daughter cells, that process is known as cell division. So what are the two main properties of a cell? It is cell growth and cell division. And also we know that life starts from a single cell, that is zygote, right? So how zygote will be formed? So zygote is formed of fertilization. Yes, so fertilization, what happens where fusion of male and female gametes are takes place. For example, if you consider this as male, okay, so this as male gamete, so this is haploid in nature. Okay, and here I'll draw, if you consider this as female gamete, this is also haploid in nature. So during fertilization, what happens is the male gamete and female gamete fuses each other to form diploid. Okay, so this is diploid zygote. Clear? So this diploid zygote grows and it undergoes division. Clear? So that is why we have more than 10 billions of cell. Okay, so human body, it is actually organ system level of organization. That means that for a digestion we have digestive system, for respiration we have respiratory system, for immunity we have immune system, and for blood circulation we have circulatory system, right? So we have organ system level of organization. So what happens to divided cell? So divided cell has to differentiate, means they perform different functions. For example, certain cells form into eyes, nose, reproductive organs, tissues, etc. So the process is called as cell differentiation. Okay, so now let us discuss with the definition of cell cycle and cell division. So the growth and reproduction of all organism, it is depend on the division and enlargement of cells. Okay, the mechanisms of division and multiplication of a cell, which is together constitute cell reproduction. Cell reproduction is nothing but it is another name for cell division. Clear? Okay, so the definition of cell cycle is it is the life period of a cell during which a cell synthesizes DNA, that is called as replication. So DNA has to replicate its own copy, and growth of a cell takes place, and it will divide into two daughter cells. Clear? Okay, so here DNA of course synthesizes, but the DNA synthesis takes place only at specific stage of cell cycle that we discuss little later. Clear? Okay. So basically, cell cycle is completed in two steps. The first phase is known as interphase and second phase is called as M phase. Okay. So this interphase, earlier it was believed that it is resting phase. So it was believed that the cells are resting at this time, and whereas in M phase it is the multiplication phase where the cell is actually divides here. So here certain events are taking place, but in interphase nothing is visible from outside. So earlier scientists, they believed that it is resting phase, but actually interphase is very active phase. So your preparation for M phase, that is all the preparation for multiplication occurs in interphase. That is why it is also called as preparatory phase. So the interphase, it is again divided into three sub-phases. Okay, they are G1 phase, or it is also known as gap 1 phase. Second one is S phase, and third one is G2 phase. It is very important. So interphase is further divided into three subphases: G1, S, and G2 phase. Whereas M phase, it is divided into two categories. One is karyokinesis and second one is cytokinesis. So karyokinesis is nothing but division of nucleus. So here nuclear division will occur, and it is completed in four parts, that is prophase, metaphase, anaphase, telophase. Okay, we will study these stages in the upcoming videos. So in these four phases, nucleus is going to divide. Here caryon term is used for nucleus and kinesis is for breaking up or splitting. So after the nucleus has divided, the cytoplasm is going to divide, right? So that is known as cytokinesis. Clear? So karyokinesis is nothing but division of nucleus and cytokinesis is nothing but division of cytoplasm. So in cell cycle, I repeat, there will be preparation phase and it is called as interphase. It has three division: G1 phase, S phase, and G2 phase. Whereas M phase is nothing but multiplication phase. Here division will takes place and initially nucleus is going to divide, that is called as karyokinesis, and later the cytoplasm, and it results in two daughter cells. Now let us discuss with the representation of a cell cycle. So it is represented in cyclic manner. You can see here. So this is the representation of cell cycle. So here you can see M phase, right? M phase, that is division phase. Then it enters into interphase. From here to here, it is called as, here it is known as interphase. It includes G1, okay, here is G1, S, and G2. So including G1, S, G2, what is going to happen here? Your preparation for division takes place. Okay, only in M phase the actual division occurs. Now we'll see different events that are taking place in G1 phase. Okay, we'll move on to next slide. Before that, so what is interphase? So interphase is nothing but it is the phase between two successive M phases. Correct? So it includes cell growth and DNA synthesis. And interphase is the longest phase because it lasts for more than 95% of the duration of cell cycle. Okay, only 5% will be the division phase, rest of the thing will be interphase. Okay. Now, as I mentioned, interphase has three phases: G1 phase, S phase, and G2 phase. Okay. So now let us discuss with G1 phase. Okay. Of course, it is the first growth phase, that is why known as G1. Okay. So it is the interval between mitosis and DNA replication. So mitosis is nothing but it is used for M phase. So in between mitosis and DNA replication, that is the S phase, there is a phase called as G1 phase. Now let us discuss with the main events which are taking place in G1 phase. Okay. Now, main thing you should remember that the cell which prepares itself for the next stage. So what is the next stage here? That is DNA replication. Now we'll recall what and all we require for DNA replication. Okay. So for the DNA replication, first thing is certain enzymes are required, right? So we will see DNA replication in detail in unit 1. So there are certain enzymes called as helicase, topoisomerase, DNA polymerase. So these enzymes are required. So we know most of the enzymes are proteins. So here protein synthesis increases. Clear? For protein synthesis, transcription and translation should occur. So for these, all types of RNA should be there, right? rRNA, mRNA, tRNA. So here synthesis of RNA also increases. The first thing is synthesis of protein increases. Second thing is synthesis of RNA is also increases. So ATP synthesis also increases. For all the events we require energy. So energy will get from ATP. So synthesis of ATP also increases. For DNA replication, raw materials are required. They are known as machinery for DNA replication, like pentose sugar, nitrogenous base, etc. So they all synthesize here. Now everything is ready for DNA replication. Now what happens? The DNA is going to replicate using all these things. So this is what happens in gap 1 phase or G1 phase. I repeat, main events are which continuous growth of a cell become metabolically active, which prepares machinery for DNA replication, and synthesis of RNA and proteins takes place. So next we will talk about what happens in S phase. So here in this, first change as DNA replication takes place. Okay, so in this DNA replication as a must event happens. Second important thing is, if it is an animal cell where centriole is duplicated or centriole replicates, but in plant cell where centriole is absent, so this point is not applicable for plant cell division, but in animal cell centriole is also replicate. So next point is histone proteins are synthesized. So when we read cell cycle from various textbooks, we find that in some textbook it is written that histones are produced in G1 phase, but in some textbook it is given that histone proteins are produced in S phase. So both these informations are not totally wrong. Why? Because lower eukaryote, it produces 50% of histone proteins in G1 phase, whereas 50% in S phase. But in higher eukaryotes, the complete or full histone proteins are produced in S phase. Clear? Now why histones are required? So these histones make octamer, and around which DNA wraps around. Clear? So that complete structure is called as nucleosome, so that DNA can coil properly. Clear with this? So this is about S phase. I repeat, in this DNA replication takes place, amount of DNA per cell doubles, but chromosome number is not increased. In animal cell, replication begins in the nucleus and the centriole duplicates in the cytoplasm. So next we will see about G2 phase. So in G2, as I said, in every stage the cell prepares for the next stage. So in G2 it prepares the cell for division phase, right? So in division, what happens? So nucleus has to divide, new organelles should be formed, the chromosomes gets separated, and spindle fibers are formed. So this is generally happens in division. So it prepares the cell for these things. Now in spindles, they are made up of a protein called as tubulins. That means a protein synthesis should takes place. Clear? I repeat, spindle fibers are made up of a protein called as tubulin. So since it is a protein, so protein synthesis should occur here. So for protein synthesis, prior to this should be transcription and translation. For that, RNA should be required. So RNA synthesis also increases, plus ATP synthesis also increases here. Now the cell is ready to divide. Clear? So I repeat, in G2 phase, it is second growth phase where cell growth continues, synthesis of RNA and proteins continues. Cell is prepared for mitosis. Okay, so next is about YUM phase. So this could be mitosis or meiosis depending upon the type of cell. So YUM phase represents actual cell division. In human cell cycle, it lasts for only about 1 hour. YUM phase includes karyokinesis and cytokinesis. As I mentioned, karyokinesis is nothing but nuclear division. Cytokinesis is nothing but division of cytoplasm. Okay, so some cells, they don't show any division. For example, heart cell. Many other cells divide only occasionally to replace damaged cell or dead cell. Now we'll see about a special phase, that is G0 phase. Okay, before that, okay, so there is a daughter cell will be formed in YUM phase, right? So diploid cell which will enter into cell cycle. So first itself it prepares for S phase. In S phase, what happens? DNA replication takes place. In G2, it prepares itself for division phase. Then nuclear division occurs, then cytoplasm. So when cell comes here, we get two daughter cells. Okay, and those daughter cells will have 2n chromosome number in mitosis. Clear? So suppose the formed two daughter cells here. So here will be two daughter cells are formed. Suppose these two daughter cells are not going to divide. That means they will get differentiated or specialized to perform a specific function. So the cells initially enter into G1, they grow to normal size, and then they get into a stage which is known as G0 phase or G0 phase. Clear? Okay. Now when this happens, so when G0 will occur, so this happens if there is absence of energy rich compound, if there is no ATP molecule which is available for division. So that time it moves into G0 phase. Here you can see G0. The G0 phase is inactive. That means that it is inactive only in terms of division. So the cell which enters into G0, which is not going to divide, but it is metabolically active, means it is going to perform its activity except division. So this stage is known as quiescent stage. Here you can see this is called a quiescent stage. Okay, so the cell come out from the cell cycle and they are not going to divide anymore. So this process is called as differentiation. But in some cells, for example parenchyma cells, there is an exception that it will come back from G0 to G1 phase again. Then they will undergo cell cycle. But in most of the cell, they will enter into G0 phase. So this is about interphase and YUM phase. So we'll recall the points once again.
This video that a cell cycle has two major phases. Do you remember the names of the two phases? That's right, interphase and M phase. we have seen that the interphase lasts longer in duration compared to the M phase. The major reason being that the interphase deals with preparation for the actual division, which occurs in the M phase. Now, in this video, let's have a look at both these phases in detail. Since these phases are quite complex, we divide them in substages to understand them better. Let's begin with the interphase first. Here is an image that depicts the complete interphase in general. As we can see, the phase has 3 stages. Let's begin each stage step by step. The first stage of the interphase is the first gap phase, abbreviated as G1 phase. This is the first phase occurring in interphase, hence the name first gap phase. It corresponds to the interval between the M phase of the previous cycle and the S phase of the current cycle. Since this is the first stage of the interphase, that is, the first stage of the preparatory phase, and the phase which precedes synthesis of DNA, what do you think will be the activities of the cell? Just think about it. Well, as the cell is metabolically active, it grows in size in the G1 phase. All the organelles duplicate in this phase, and the building blocks which will be required in later stages are made by the cell. It is important to note that the G1 phase does not involve DNA replication. Now let's move to the second stage of interphase, which is the synthesis phase, abbreviated as S phase. As the name suggests, there will be synthesis of something. And what will that something be? The most crucial part of each cell, that's the nucleus, along with the genetic material within, will get synthesized, that is, duplicated. So the S phase takes up the charge of the DNA replication. Now, along with the nucleus, there is one more important organelle that gets duplicated in the S phase. Do we know this bundle of threads present in the cell? That is called the centrosome. This bunch contains smaller organelles called the centrioles placed within, usually at right angles. They are made up of microtubules which help in releasing the threads required for separating sister chromatids later. We will have a detailed look at this process in our upcoming videos. For now, all we need to know is that these threads are important for the cell division. And for this reason, duplication of the bundle containing these threads is also important. This happens in the S phase. So the S phase takes up the responsibility of duplicating the contents inside the nucleus and the centrosome present in the cytoplasm simultaneously. What next then? The next step in the process of the interphase is the second gap phase, abbreviated as G2 phase. This phase allows the cell to grow more. Along with the enlargement in the cell size, the cell also makes more proteins and organelles. The most important function of this phase is that it prepares the cell for mitosis. This phase helps the cell to prepare for the actual cell division in the M phase. So now, are we set to study the next phase? That is the M phase? No, not yet. We are yet to study one more intermediate phase. A stage which can be entered post the M phase of one cycle and before the G1 phase of the next cycle. There is a phase called the G0 phase in between. So why didn't we talk about this phase in the beginning before the G1 phase? Well, the cells may or may not opt to enter this phase. The cells may enter the G0 phase or may directly enter the G1 phase after the cell division. And what factor decides this? Well, there are several intrinsic and extrinsic factors like resource availability or nutritional deprivation which can force the cell to enter this G0 phase. But what exactly happens in this phase? This phase was classically thought to be only a resting phase, as the cell is metabolically active but does not show tendency to divide further. However, recent studies suggest different names for this phase. If the cells enter the G0 phase such that they returned to G1 phase after some time, then it's called the quiescent stage. That means the cells will be metabolically active but only arrest its further division. However, They can re-enter the G1 phase as and when possible. So this kind of phase where the G0 phase can be reversed to enter G1 will be the quiescent stage. But if the cells enter the G0 phase irreversibly, then they're said to enter the senescent phase. That means the cells will only be metabolically active but never show the tendency to divide further. Hence, this phase is called senescent phase, the one from where there is no return towards further division. So depending upon the extrinsic and the intrinsic factors, the cells enter the G0 phase, which is a metabolically active phase. The only difference is that the cells do not divide further either for some amount of time or permanently. So G0 phase is the phase where the cell division halts, keeping the cell highly active otherwise. Now that we've had a look at all the important phases in the interphase, let's have a look at the next important part of the cell cycle, that is the M phase. This could be mitosis or meiosis depending upon the type of cells. To our surprise, even this M phase shows different stages.
Hope you understood the video. In the next video, we will learn about different stages of mitosis, that is prophase, metaphase, anaphase and telophase. Okay, thank you.